T-cell receptors to sort of dissect and learn about our immune response. So I'm not touching about on vaccine design or so on, but we will focus here mostly on the methodology part. But before we start, I'd like to give you a quick whirlwind tour through T-cell immunity, specifically the T-cell receptor, so that you can appreciate the later talk as well. Okay. So the T cells are one of the main component of our adaptive immune response, and they are responsible of identifying and sort of destroying pathogens that invade our human body or cancer cells. And the mechanism they usually use to do this is through a recognition of so-called antigens, also, which are essentially either of pathogen origin or alterated by cancer through their T-cell receptor. And these antigens, or a small fragment of these antigens, so-called epitope, is presented on a surface of cells by the so-called surface expression molecule called MHC. And if a T cell can then recognize this expressed or presented peptide through their T-cell receptor, they will activate and then induce an immune response that ultimately leads to the destruction of the origin of this antigen. So to understand how this is presented, we need to sort of look into the three-dimensional structure of the MHC molecule and the T-cell receptor itself. So these peptides or antigens are linearly presented to the T-cell receptor, bound to these MHC molecules. And here we distinguish between two classes, namely MHC class 1 and MHC class 2. So where MHC class 1 has a very well-defined binding pocket, which looks more like a hot bathtub, is closed and therefore very restricted of what kind of length of peptides can be presented and bound there. MHC class 1 is much more open and more compared to a hot dog bun, where the peptide still lies linearly in the binding groove, but can sort of dangle around like an actual hot dog. And so the T-cell receptor itself interacts then with this complex. And if a strong enough interaction happens, the T-cell will get an activation signal that then leads to an induction of an immune response. And here, the three key components play a crucial role, namely the CDR3 or CDR regions of the T-cell receptor. Here, the T-cell receptor itself is a heterodimer consisting of an alpha and beta chain, and both have three different loops that usually interact with the complex, the MHC peptide complex. And here, specifically, the so-called CDR3 region is supposedly in contact with the peptide. And so this is then determining whether a peptide or whether a T-cell receptor is really recognizing a peptide and therefore inducing or gets activated. Yeah? And so the key point here is, if we want to understand when a T-cell is activated, we need to understand this interaction. Yeah, and one major aspect here is that the T-cell receptor itself needs to be highly diverse to be able to really recognize the plethora of different pathogens that are out there. And the way our immune system does this is through genetic recombination, a so-called VDJ recombination, where the T-cell receptor itself is encoded by multiple different segments, genes, the VDJ genes, that are then recombined during maturation. So, for example, the beta chain here consists of V, D, and J recombination, and usually first the D and J elements recombine, and then the V element recombines, and together join then to a beta chain. And usually this is also the diversity that comes through these recombination can be further enhanced through sort of sloppy joining by integrating random base pairs at the joining elements. And usually exactly there, these parts are encoding the CDR3 region of the T cell receptor. And so it's not surprising that in most cases, the CD3 region is determining the binding specificity of the T cell receptor. There are, of course, exceptions to this, where other CDR regions are also determining the binding affinity of the T cell receptor, but in the majority of cases, this is mostly determined through the CD3 loop. So this is then also done on the other side. The alpha chain is also then recombined similarly. However, here we don't have a D region or D segment. And together, through these recombinations, we can hypothetically create 10 to the 21 possible T cell receptors. And so you can imagine this is the huge design space that we, or the huge sequence space that we are here expressing. Is quite demanding then when you tackle this through machine learning or data-driven approaches. But generally, we have only 10 to the 7 or 10 to the 8 different T cell receptors at any moment circulating, and that ensures that we can recognize almost all pathogens that we can encounter. So now the general question is, can we sort of model these interactions, although we have this high variability in our data, to be able to predict for a given T cell receptor and a given epitope, whether this epitope can be recognized by a T cell? Yeah, so generally, the determining factors of an epitope aren't really clear, yeah? And data-driven approaches up till now were hampered by a lack of existing paired data. So the usual approach people took is essentially extracting these interactions based on co-crystallized structures and then training models based on this information. And of course, you can imagine these experiments are quite demanding, very low throughput, and therefore we up till now didn't have a lot of training data for the data-driven approaches. This has changed quite recently due to new high throughput experiments that allow us now to really sort of probe the entire epitope space for a specific T cell receptor, or vice versa, sequence determining the entire specificity landscape of a repertoire of T cells for predefined epitopes. And so with these new high throughput experiments, we can now generate within one experiment as much data as we collected over the last 10 years or so. So there's a huge influx of data, which makes it now at least approachable through data-driven approaches. And so this is essentially what we tried to do in our first project here in collaboration with the TICE lab led by David Fischer, where we wanted to exploit these newly single-cell experimental procedures that can now sequence an entire T-cell repertoire of a tissue while probing the specificity of that repertoire for a predefined set of epitopes. So we can sort of probe up till a couple of hundred peptides. For a couple of hundred peptides, the specificity of the entire repertoire of the tissue. And so this usually creates data points of like 90,000 different T cell receptors in that specific data set, for example, that we used to train this model. And so we approached then this through, through a deep learning architecture here, where we encode the alpha and beta chain through a sequence encoding stack, and then combine the latent representation of these alpha and beta chains together with additional covariates that sort of account for the cell state, but also for certain peculiarities of the single cell experimental protocols into a normal feed-forward neural network. And then we have a multitask output here representing each individual epitope that was part of our experiment. And the task here is then to predict for an unknown T cell receptor whether it can recognize any of the epitopes that were part that we're specifically and explicitly modeling here. And it turns out in this very refined and reduced problem setup, we can achieve quite high prediction performance. And what you can also appreciate here is that generally, if you combine the alpha and beta chain, you have a slight increase in performance. but usually the alpha or beta chain already suffices to reach quite good prediction performance. Peculiarly here, though, is that we already get quite high prediction performance with simple linear embedding layers or convolutional embedding layers. So this means we can sort of reduce this problem in this setting to a modifying problem within the alpha and beta chain. Of course, however, this architecture is extremely stringent, right, and very rigid in terms of what we can do with this. We can use this perhaps to stratify patients, for example, for cancer vaccine designs that specifically target an individual epitope, but we cannot do any kind of generalized predictions with this architecture. So the next question, of course, is then, can we now, with this influx of data, can we generalize to unseen T-cell receptor and epitope pairs? And to that end, we sort of added on to this architecture here a second embedding layer that now takes into account also the peptide sequence, so the epitope sequence itself, and then combines both T-cell receptor and epitope sequence in a feed-forward layer to do then a binary prediction whether the specified T-cell receptor here can recognize the specified epitope sequence. And so when you then test this generalized architecture on T-cell receptors that recognize an epitope that were part of the training data, this generalized model was usually outperformed by our previous categorical model. Yeah, this hints at that already this generalized model couldn't really extract sort of general rules of interaction between the T-cell receptor and the epitope. And this becomes unfortunately very painfully clear if you do out of sample predictions here. So training the model on an entire data set from a different database and then predicting on a different data set entirely. And here you see that this model essentially didn't learn anything and is a random predictor, which is not surprising if you consider, although having lots and lots of T-cell receptors, we only have paired information of the T-cell receptor and epitope space of a couple of hundred epitopes. And this is clearly not enough to approximate and generalize across this epitope space. Yeah? So we need here much more sophisticated models and models that perhaps take into account structural information or so on to be able to do any kind of generalized predictions. So since we are not able to generalize to unseen pairs, we asked ourselves, can we do at least predictions for slight perturbed peptides for the same T-cell receptor? And to that end, we created an alterated peptide ligand library for a very specialized epitope sequence, the synfactal sequence. This was done in our collaboration partners lab, Schubert lab and Bush lab. And then Emilio and Felix, two PhD students in my group, tried then to model this type of mutational data to be able and ask the question whether we can predict whether an T-cell receptor or a T-cell is still activated when challenged with this alterated peptide. Yeah? And so our collaboration partners essentially mutated this peptide at any position with all different amino acids and then measured the activation of these T cells or T-cell repertoire that was first predisposed to the wild type sequence and a naive repertoire. Yeah, and sort of to and then measured whether they have been activated or not. And so this data set then consists of roughly 6,000 data points of roughly 38 different T-cell receptors, some naive, some educated. And then based on that, we trained a random forest model to do two tasks. One, to predict within a T-cell receptor the binding affinity, well, the activation score, I should say, and a classification whether this T-cell is sort of activated and can be recruited to the tissue. And then another setting where we try to do this across T-cell receptor sequences. Yeah. And to this end, we encoded the peptide sequence here through dimensionality reduced physicochemical properties, and then not only encoded the sequence itself, but also the wild type mutation, the position, and the wild type amino acid at that position, and the mutation, and entirely the entire positions that have been mutated. And then through that encoding, we then trained this model. So to give you an idea of how this data looks like. So on the one side here in orange, we see the activated or educated repertoire. And you appreciate here that generally we see a stronger activation in that educated repertoire versus this naive repertoire, because this has been essentially pre-exposed already to that epitope. And if we look also just into the individual contributions of the positions in that epitope, we appreciate also that certain positions seem to be more robust to mutations, whereas others reduce quite dramatically the activation score. And this is especially here, position 2 and 5 and 8, for example, seem to be very robust. And this is very much in line with what we know about this peptide, because we know that these positions are sort of anchor points and are buried into the MHC molecule. And so they are not exposed to the T-cell receptor. Whereas position 4, for example, is known to be in interaction with the T-cell receptor. So then we trained our random forests here, and then looked into how well we can predict essentially these activation scores and recruitment scores within that same T-cell receptor. And generally what we observed is that we can, for the educated repertoire, achieve quite nice predictions. So usually in a 90% AUC regime, and also the got very nice spearing correlations to the activation score. And this also sort of carried over to the next task, where we try to do predictions across T-cell receptors. And here, generally, again, educated repertoires could be nicely predicted, whereas naive repertoires, it was much more diverse. And here we could see trends, specifically those T-cell receptors in the naive repertoire that had a high activation generally could be predicted quite well, whereas others that didn't activate strongly behaved very erratically and therefore couldn't really be predicted quite well. So of course, given that there are already existing models that claim to be able to predict activation for or specificity for an unknown T-cell receptor and epitope pair, we were curious whether they can also predict these very minute alterations. But it seems that these models do not really predict this specific task quite well, were always outperformed by our model. So then we asked, what did the model actually learn? Did it actually extract any kind of biological knowledge, or did it just learn random information out of the data? And to that end, we looked into feature importance through perturbations. And here specifically saw that if we exclude the CDR3 in the model, then the performance dropped significantly, which is not surprising because you need that information to sort of go beyond an individual T-cell receptor. And then if you remember the plot from before, we see here as well that position 4 in particular has also quite importance to the prediction specificity prediction quality. And this is not surprising given that we know that this interacts probably with the T-cell receptor sequence. And so to verify this, we then started modeling these APL libraries through crystal structure modeling, 3D modeling, and here could also verify that especially these positions that seem to be important for prediction quality are usually also in close proximity to the CDR3 loop. And here just two examples of two highly, so the two left are examples of highly affine T-cell receptors that activate quite strongly. And here on the right, you see an example that doesn't activate well, and you usually have higher distances between then the T-cell receptor and the epitope itself. So it seems like our model did truly extract functional and structural aspects of that data, and therefore was able to really gain these prediction qualities. So what we also experienced is that we can actually reduce the amount of training data quite significantly without reducing the performance dramatically. And so indeed, we can reduce the amount of training data by like 50% without dropping drastically for the educated repertoire. And it also seems like it doesn't really matter whether we leave out certain amino acids, as long as they're distributed evenly across positions. But if you remove an individual position, then the performance will drop drastically. And then, of course, it depends on which position you're removing from the training data. But this overall means that we can drastically reduce the experimental burden as well. So to sort of guide this, we then turned to active learning and used then the random forest class probabilities to select essentially the next experimental point to verify. And what we can appreciate here is that we indeed can, with this active learning procedure, reduce dramatically the amount of experiments over random selections. And especially if we're interested in sort of regressing the activation score, we see that usually this performs quite optimally, and we're usually reaching sort of the greedy upper bound quite sufficiently with this active learning procedure. Just to clarify, this upper bound is sort of looking back and selecting greedily the next best mutation or data point that would increase optimally the performance of the model. So this is a greedy upper bound here. Okay, so for now, so far, we only looked at a T-cell receptor sequence, but there's more to a T-cell activation than just the sequence itself. Yeah, so a T-cell can be in different states, and depending on what kind of transcriptional state they are in, they will react differently. So a T-cell can be, for example, exhausted, which means that even a recognition of an epitope won't activate the cell. And so, this means we need to sort of combine the cell state and the T-cell receptor in order to truly model the behavior of a T-cell. And this is what we try to approach through this experiment here, or through this project, which is also in collaboration with the TICE lab and done or spearheaded by Felix and young PhD students in my group. And here we tried to build an multi-view conditional variational autoencoder to integrate jointly measured single-cell transcriptomics and T-cell receptor sequences into a latent space that we can then use for various downstream tasks. For example, querying an atlas with this latent space to impute, for example, T-cell specificity, doing predictions, and generally hoping that this latent space is much more informative and therefore can then find certain aspects of the data that we would otherwise overlook if we just individually look at the T-cell receptor or the single-cell RNA-seq information alone. So to verify that this works, to do this, we then tested multiple different mixture models that combine essentially the latent representation of the transcriptomic space and the T-cell receptor space. And here we tested 3 different models, namely a simple one where we concatenate essentially latent encodings of the 2 modalities and then feed that again through a feed-forward neural network to get a joint latent embedding. But then also other factorizations like the product of expert factorization or a mixture of expert factorization. And sort of to quantify whether these worked and are actually better and add something to the interpretation of data, we defined different matrices. So one metric is whether the specificity of our T cells, which is not specifically encoded into the information here, is recovered in our joint latent space. And what you can see here on the left is the latent space representation represented here as a UMAP, and then the 2 unimodal embeddings. And what you can appreciate here is that, yes, the specificity seems to be better represented in our joint model versus the RNA's only model, of course, because the RNA data doesn't have that necessary information of the T cell receptor sequence. Whereas in the T cell receptor sequence, we have a perfect separation, but usually very fine granular clusters. Yeah? Similarly, we asked whether we find whether the clonotype, whether clonotypes are conserved in our joint latent space. So clonotypes are essentially groups of T cells that have the same T cell receptor or highly similar T cell receptors. And here we're plotting essentially the most abundant clonotypes. And as you can see here, they are again nicely grouped in our latent space, in our joint latent space. RNA, not so much, and T cell, of course, they are. So again, these both informations are still contained in our joint latent space. And lastly, then, sort of to confirm that also the transcriptomic space is present, we looked into then a cytotoxic score that essentially is defined by a multitude of marker genes. And here we saw essentially that our general latent embedding is sort of better refined and shows a hierarchical separation of these expression patterns, whereas in a T cell receptor space where we don't have that information available, we see essentially a random pattern. So that overall leads us to believe that our latent embedding seems to combine both information and represents them at least in a concise manner. And of course, that can be also quantified. And to that end, we specified again a multitude of tasks. First of all, we wanted to predict essentially the specificity of an unknown T cell receptor. So which epitope it reacts to based on k-nearest neighbor latent embeddings. Yeah? And compared this then to essentially T cell receptor only and RNA only embeddings. And here, generally what we observed is that we can reach higher predictive performance with our joint latent embedding versus our single latent embeddings. And similarly, we wanted then to see whether quantitatively also the cell types are still sort of contained in our latent embedding. And to that end, we used a normalized mutual information as a criteria with predefined cell type clusters based on RNA space. And here also we could show that generally this is better conserved than if we look into unimodal, and same holds true also for reactivity, which is defined by the T cell receptor and cannot be captured by the RNA space. So lastly, then we looked into a competitor method here, and it's called TESSA, which is also combining both RNA and T cell receptor information to do a Bayesian clustering. And here again, we use then their distance matrices and to do latent space or to do k-nearest neighbor embeddings to see whether they can sort of conserve specificity in their space. And it seems that our model does a better job than TESSA. not just in specificity, but also in conserving cell type, clustering, and reactivity clustering. So overall, it's a richer representation that allows us to do, to represent the data more faithfully. So then with that, I'd like to very briefly tease you a bit. So this is an ongoing still analysis. So here we looked into a very large single cell data set consisting of multiple different tumor tissues and the T cells within. And this is roughly 500,000 cells integrated here. And I'd just like to appreciate here that the separation of these tumor types and tissues seem to be much more nicer conserved and clearer separated in our joint latent embedding on the right, whereas in a TCR, whereas in a transcriptomic space, we see more intermingled, less defined clusters forming. So with that, but this is still ongoing, and we're doing essentially analyses on top of this right now. And I wanted to just tease you with that. And you need to wait, unfortunately, for the next presentation here, or our paper that hopefully comes soon out. So with that, I'd like to thank you all for your attention and happy to take questions.